behalf of the IQ Consortium and the Translational and ADME Sciences Leadership Group, I would like to introduce our speakers for today's working group webinar, Drs. Yen Chen and Tamara Kabalu. Dr. Chen is a principal scientist in the Department of Drug Metabolism and Pharmacokinetics at Genentech. She has 20 years of experience in DMPK discipline and an expert in physiologically-based pharmacokinetic modeling, or PBPK modeling, for the prediction of human PK, absorption, and CYP and transporter-mediated drug-drug interactions. She leads the PBPK effort at Genentech, overseeing PBPK support to all projects. Dr. Chen has been an active member on IQ PBPK expert working groups, including leading this Itraconazole PBPK working group. Due to her dedication, outstanding contributions, and leadership of this working group, she received the prestigious IQ Recognition Award in 2019. Our second speaker is Dr. Tammy Kabalu, who is currently a principal scientist in the Department of Pharmacokinetics, Pharmacodynamics, and Drug Metabolism at Merck Research Laboratories. She has a Bachelor of Science degree in chemistry from Bradley University and a PhD in bioorganic chemistry from Washington State University. Since joining Merck in 2007, she's supported preclinical ADME activities for numerous programs from discovery through development. She co-leads the department objectives focusing on development of PBPK scientific strategy, and she's been leading the SimSip super user group there within Merck for nearly 10 years. And I want to thank you all in advance, particular speakers for this presentation and attendees, and I'll turn it over to our first presenter, Dr. Chen.
Thank you, Heidi. Thanks, everyone. So as Heidi introduced, so Tammy, will you forward the slides? Yeah. So I think in 2016, we started as the IQ working group focused on Itraconazole PBPK with the mission of providing the recommendations sorry, for the optimal Itraconazole DDI study design. So our goal is using the newly generated in vitro data and collected the clinical data to develop the model with better mechanistic understanding so that we be able to use the model to simulate various clinical DDI scenario to provide the recommendation on the optimal study design. So the working group comprised members from 14 companies with expert in the PBPK field, as well as the members with experience on the clinical pharmacology. So as a product, we have published our work in the CPT PSP, and so people, if you haven't had a chance to read it, would like to know more details, you can find the paper there. So today, Tammy and myself would like to share some details of our work. I will start with a very brief introduction on the objective, our strategy. Tammy will go through the core part for the model development verification. Then I'll close the talk briefly talking our recommendation and share with you a couple points of discussion. So as most of you are aware, due to the hepatotoxicity, ketoconazole has been eliminated or can no longer be used for the clinical DDI study to test the CYP3 substrate. Based on the literature search, I think in 2015 or 16, CPLG have recommended a best practice, but in that work they did recognize that consider the complexity of this Itraconazole DDI, the PBPK simulation may play a more important role in supporting or confirming the study design. So at that time, there has been Itraconazole model available including some commercial available software. But I think the consensus is from all of us is we believe to develop a mechanistic model, it is quite challenging due to several reasons, such as more complex PK, due to you have not only the inhibitory, itraconazole, also the inhibitory metabolite, as well as the challenging to generate or obtain more reliable in vitro data due to the drug property, both for the parent and the metabolite. So there is a desire from both pharmaceutical as well as regulatory to see the recommendation of the DDI study, especially through mechanistic PK simulation. So here's a brief, our overall strategy. Tammy will go through details for each of the content in this box. So very briefly, I think this working group actually generated in vitro data through the lab in our member company. And we also collected the itraconazole and the metabolite data that's from a more recent clinical trial in this member company. So for the model development, we started to and focus on the bottom-up approach. And so for the model verification, again, we used the itraconazole data collected from the member company. For the DDI verification, we used the 20 clinical studies, including midazolam as well as a couple other CYP3 substrate. Then used the verified model, did the simulation to look at various clinical scenarios. So before we go into the details that Tammy will talk, I just want to really highlight a couple of key challenges that we have been worked on and considered. As I mentioned, there has been itraconazole tape model available. People have lots of learning experience from there. So in our work, we have focused on a couple of points. First, again, this in vitro data, right? How the acid condition could impact our interpretation when we put those parameters into the model, and what is the consistency of those data across the lab, and also which metabolites we should include, just major or all of them, because we do hear there's a different lab and scientists have probably different observation and opinion. Also, we try to also look at whether the absorption model could impact the prediction of the DDI, since we understand different absorption model may use different interacting concentration. And lastly, as I mentioned, even we try to focus and start with a bottom-up approach, when those translation becomes not a one-to-one, and instead of just fit the parameter, we tried some hypothesis-driven study to convince us and to rationalize why we need to scale that, and so on. So without further on this, I'll transfer to Tammy to go through more details. Go ahead, Tammy.
Thank you, Yen. So as Yen mentioned, I'll get into the model development and verification steps. So the working group started out by collecting clinical data to support both model development and verification. This included PK data as well as DDI data. PK data from the literature were used in model development, and then the data collected from the working group member companies were used in the model verification step. So the data used in the model in development included IV data at several dose levels and single and multiple oral dose data using both the fasted, the oral solution as well as the capsule formulations. Data included in model development were 11 oral solution studies, most of them after multiple dosing, and 6 studies from the capsule in both the fed and fasted states. And most studies collected from the working group reported concentration time data for both itraconazole and hydroxyitraconazole. And then we had 6 additional studies that had data for the 2 additional circulating metabolites, keto-itraconazole and N-desalkyl-itraconazole. Clinical DDI data was obtained from the University of Washington Drug Interaction Database, in which we searched for trials that had an in vivo inhibition effect of over 20%. And then that list was narrowed to CYP3A4 substrate that had a model already available in SimCYP. So then we ended up with 20 trials with 7 different victim drugs of varying fractions metabolized by CYP3A4. So in parallel to the clinical data collection, the working group generated a robust data set of the key in vitro parameters required to support model development. And this included data on physical chemical properties, plasma protein binding and binding in human liver microsomes, CYP3A4 enzyme kinetic and inhibition data, and hepatic uptake. So over the next several slides, I'll go over the experimental results of these data, and then we'll get into how they were used in model development. The physical chemical properties assessed by the working group included log P or log D and pKa for itraconazole and its 3 metabolites, and then permeability and solubility at different pHs and in biorelevant media for itraconazole. And overall, we thought that the values we obtained were what we anticipated based on previous reports, and were in line with itraconazole being highly lipophilic, weak base with pH-dependent solubility. Plasma protein binding data were generated by the working group for itraconazole and its 3 metabolites using equilibrium dialysis. And our values were all within about 2-fold of what was reported by Riccardi et al. In 2015, also using equilibrium dialysis. And then in addition, our value for itraconazole was very similar to what was reported by the IQ working group on plasma protein binding in 2017. So taken together, these 3 separate efforts indicate that itraconazole is about 10-fold more highly bound than the historically reported value, which used a tube absorption method rather than equilibrium dialysis. So CYP3A4 inhibition data was generated by the working group in human liver microsomes, and that free fraction in the microsomes was assessed using equilibrium dialysis. And what we found was that unbound IC50 values for itraconazole, hydroxyitraconazole, and keto-itraconazole were pretty much in the same range as the previously reported values, then about 1 to 4-fold. However, in our hands, the unbound IC50 for N-desalkyl-itraconazole was about 40-fold less potent than what was previously reported, with 19 nanomolar compared to 0.4 nanomolar. And this difference was found to come from the difference in the FU-MIC determination, in which we found N-desalkyl-itraconazole to be a lot more free than what was in the literature. And it's likely due to the differences in methodology, with equilibrium dialysis being used by the working group and the tube absorption method being applied in the previous value. And the value we generated for this metabolite is actually in line with its physical properties and its log P relative to itraconazole itself, where you'd actually expect it to be slightly more free in microsomes compared to itraconazole. So we use these data to assess which of these metabolites contribute to the overall inhibition of itraconazole, and ultimately decide what metabolites to include in the itraconazole model. So the in vitro KIs were calculated from the unbound IC50 values we generated and compared to in vivo unbound plasma concentrations from the PK data collected by the working group. Sorry. It's not letting me get to that slide. Hold on. Sorry, I can't zoom into that slide for some reason. so it's going to look really small. But what this plot shows is the unbound concentrations over time for itraconazole and its 3 metabolites after a 200 milligram QD dose of the oral solution on day 4. So you can see the itraconazole and hydroxyitraconazole, the 2 plots on top, the unbound plasma concentrations are at or significantly higher than their unbound Ki values for a decent amount of time. However, with keto itraconazole and indazalkyl itraconazole, the unbound plasma levels are a log order or more lower than their in vitro Ki values, suggesting they're unlikely to contribute to an in vivo inhibition effect. And so therefore, with this analysis, the working group decided to not include keto and indazalkyl itraconazole in the itraconazole model, and we built the model for itraconazole and hydroxyitraconazole only. So now that I've covered the results of the experimental values, I'll now get into model development, starting with absorption models. Absorption models were built for the oral solution and the capsule in the fed and fasted states, and we constructed both a first order and an atom absorption model. The fraction absorbed and rate of absorption were parameterized using a top-down approach where we used clinical data to generate those values. The atom model was initially constructed bottom-up with in vitro data generated by the working group, but we found that the simulated AUC underpredicted the observed exposure and decided to apply a scalar to the effective permeability to recover the observed data. And then in addition, gastric emptying time was adjusted to describe the food effect with the capsule. The steady state volume and distribution was predicted bottom up for itraconazole and hydroxyitraconazole using tissue composition equations available in SimSip using the tissue composition equations available in SimSip with the in vitro data generated by the working group. And we found the value for itraconazole to be in good agreement with the clinically reported GSS. SimSip's minimal PBPK model was then used to capture the multiphasic distribution profiles for itraconazole and hydroxyitraconazole, with K in, K out, and VSAC determined by best fit to IV data. Elimination for itraconazole and hydroxyitraconazole were parameterized using in vitro enzyme kinetic data determined in human liver microsomes by the working group and assigned to CYP3A4. We found that the in vitro data in this bottom-up approach underpredicted the observed IV clearance for itraconazole, and after considering several possibilities, we decided to apply a hepatic uptake scalar to recover the observed data. And we chose this option really because it was supported by in vitro data and in vivo data in rat, what was generated by the working group. And then finally, the elimination of itraconazole was completely assigned to the formation of hydroxyitraconazole, such that its elimination served as the formation rate of the metabolite. CYP3A4 inhibition was incorporated into the model by conducting a sensitivity analysis in which we used the various unbound Ki values determined from different sources. So the working group had generated CYP inhibition data in human liver microsomes using both midazolam and testosterone as probe substrates, and in hepatocytes suspended in plasma using midazolam as a probe substrate. The simulations using the values in microsomes with midazolam were found to best capture the accumulation of both itraconazole and hydroxyitraconazole after multiple dosing. So now that I've covered model development, I'll now move on to model verification, and specifically the accumulation after repeat dosing. As I mentioned at the beginning, the performance of the itraconazole model was evaluated using the PK data collected from the working group. For this, we had information on dosage form, dose regimen, and food status available for all studies. Age, gender, and body weight were only available for some studies. And the simulations were then conducted in SimCip version 16 using the healthy volunteer population with 10 virtual trials, 10 subjects each, 50% female. So this figure shows the oral solution model, and the top plots, A and B, are the simulated PK of itraconazole and hydroxyitraconazole after a 200 milligram QD dose of the oral solution. So you can see it's overlaid with several data sets from the member companies and captures the observed data, is able to cover the observed data and accumulation well. C and D represent just a slightly different dose regimen, and that's 200 milligrams BID on day 1, followed by 200 milligrams QD. And so then the second figure is simulations using the atom model for the capsule compared to the first order model for the capsule. So E and F represent the simulated itraconazole and hydroxyitraconazole PK after a 200 milligram QD dose of the capsule in the fed state using the atom model. And that's overlaid with 4 sets of observed data for itraconazole, 2 of which had reported data for hydroxyitraconazole as well. And then G and H are the same sets of observed data and the same dose regimen, but this time using the first order model. So you can see that they both capture the accumulation well, and there was very little difference between the 2 different absorption models. So overall, the accumulation was reasonably described by both the oral solution and capsule models, and little difference was observed, as I mentioned, between the 2 absorption models. So then moving on to the second step of model verification, which was to evaluate whether a model could capture the clinical DDI data. And then as I mentioned in the data collection portion, we used clinical DDI data from the University of Washington database. And this time, again, the simulations were conducted in the healthy volunteer population, but this time the age range and fraction of females were as reported for each individual DDI study. There was no mention of dose staggering. It was assumed that the drug was dosed simultaneously with itraconazole. When dosing in the fed or fasted states was not mentioned, the fasted state was assumed. And then upon completion of these simulations, we used guess criteria to assess model predictability. So these are the results of our DDI simulations. The plots are the predicted versus observed AUC and Cmax ratios. And the dotted black line represents the 2-fold boundary, with the purple line representing the guess criteria upper limit and the blue line representing the lower limit. For the AUC ratios, 22 out of 24 were at or within the guess criteria, and there was only 1 trial just outside the 2-fold boundary. For the Cmax ratios, 18 out of 19 were at or within the guess criteria, with no DDI beyond the 2-fold boundary. So with that, the working group concluded that our model was sufficient for prospective predictions with CYP3A4 substrates and used it to inform clinical DDI study design. So I'll now turn it back over to Yen to present that over the next several slides.
Thank you, Tammy. So let's look at the next slide. So using this validated model, we simulated the various clinical scenarios before we be able to recommend the optimal study design. So with the input from the clinical pharm folks here, we mainly tested 4 things. First, to try to look at how the total duration of itraconazole administration will impact the DDI simulated. So the goal is in the clinical study design to reach the maximum inhibitory effect. The second thing, we're looking at how the substrate administration date relative to the itraconazole could impact the DDI. Third, we look at the timing of substrate administration, how that could impact the simulated DDI. And last is what would be the contribution or impact from the substrate half-life perspective. So the next slide here just showing you simulation results. I know this is quite a busy slide, but briefly, so the left figure here is identical design of the simulation. The top is for solution and the bottom for capsule. So what here basically showing you a couple of things. One is the we found from the simulation is the longer duration of the itraconazole administration, either pre or after administration of a substrate, definitely will give you higher effect. But when you close to 4 or 6 days, you pretty much reach what you get. Of course, this is also depends on the dose. And if we give the loading dose like a 200 milligram BID or 400 milligram QD, we be able to reach the highest DDI AUC ratio. And also you can see you'll be able to reach that much sooner than other QD dose. This top graph here showing you how the timing of midazolam administration with respect to itraconazole could impact the DDI, and the triangle is for the solution, and the circle is for the capsule fat. But overall, the simulation show that the timing of the midazolam administration, for example, simultaneous versus 1 hour or 2 hours later, doesn't make significant difference. If you look at closely, for the solution, looks like a simultaneous dose give you the highest number of the AUC ratio, but the difference is, you know, if it's 1 hour after, it just has a small difference. Versus for capsule under fat condition, 1 hour post will be seems to give you a bit higher, but again, the difference is minor. So this bottom mainly show you how the substrate half-life will impact the DDI where you simulated. In general, if you have the substrate have a longer half-life, you do need to dose longer post the substrate administration. So overall, based on those scenario simulation, we recommend with highlighted the 4 key points. So in general, longer administration should be at least 3 days or longer prior to the substrate dosing will definitely increase the DDI effect you will see. However, that need to be balanced with the total time by considering the half-life of substrate, because the total day of dosing for itraconazole from safety perspective, 14 days would be usually the longest. In general, the run-in period of 3 days would provide a reasonable inhibitory effect on the average. This is what's in the CPLT recommendation. The loading dose, what we observed from simulations, the 200 BID or 400 QD on day 1 could achieve highest inhibitory effect, which could be more useful for if you have a substrate that have a really long half-life. But of course, that must be used with caution for safety reasons. So the dose staggering, again, simultaneous versus 1 hour different in general, not significantly different, you will expect. And also the itraconazole dose should need to cover 4 to 5 half-life post-substrate administration in order to reach maximum interaction. So that's especially need to consider for the substrate with a long half-life. So before close the talk, I have a couple points for discussion, which something that we learned, we like to share, and some, especially from the manuscript during the expert review, we get some good questions. So again, here's one thing we want to emphasize again is there is an existing Itraconazole model. So what has been not sufficient? Where's the gap? And what's the uniqueness of this current work? So as Tammy already highlighted during her talk, so we definitely know there's gaps in the existing model, such as those questionable in vitro data and physicochemical property data. As Tammy already mentioned, the neurally generated protein binding data, in general, the free fraction would be 10 times lower than the previously or used in the model. We know those parameters will impact many things, your predict volume, clearance, and the free drug concentration. For the DDI, in this case, even could be the accumulation. And we see the need for a better understanding of those mechanistic model, such as the overprediction of the volume and also underprediction clearance, and how the in vitro Ki value that from different sources can be used, and in fact the prediction of affected the prediction of accumulation. And we want to have better understanding how the absorption model could impact the PK and the DDI. So the value or uniqueness of current work includes, again, we have carefully assessed how those in vitro assay and the in vitro data itself could impact the model interpretation and the mechanism understanding. We have collected the neurally generated itraconazole and their metabolite data from 12 companies. And while we are using the bottom-up approach, we definitely realize the disconnection for IVIVE, such as the clearance, even actually the in vitro data prediction is just a little than twofold lower than what lower than the observed. However, we tried to get a more mechanism understanding before we actually apply the FET. So the next point, Tammy also already touched upon about the absorption model. So the initial thinking is maybe the mechanistic model could have more accurate prediction on DDI, especially mostly for the gut part, because people familiar with the SIMSIP software, the drug interacting concentration for the DDI in different absorption model could be different. When you use the first order absorption, they're using polar vein concentration, whereas the atom mechanism model, you will use the interacting concentration for DDI is the enterocyte. However, at the same time, we realize in order to test that, both inhibitor and the substrate need to have an atom model. So with the help from a SIMSIP, we tested, they actually sampled a midazolam model with the atom as the absorption. So we tested that effect by simulate using different medicinal model. For this case here, we don't see the significant difference. So our hypothesis is because itraconazole is such potent inhibitor, its inhibition effect probably already reached the maximum. So, however, at the same time, we do realize, at least at that time, when we're using the version 16, that's almost not feasible, because when you have both substrate inhibitor with atom model, and especially with the itraconazole, you also have an inhibitor, the running time is like a super long. So that's our decision. In the final work, such as DDI simulation, we mostly use the first order model, but we did verify that no matter use first order or atom model for the itraconazole, as long as you simulate the concentration for metabolites and inhibit itraconazole and the metabolite is the same, then the DDI effect, which you see, will be the same. So the last one here is a comparison of the DDI prediction, compare the working group model with the existing SimSip model. I just want to point out, this is not the purpose of this whole work, as you already see the scientific part, how we do that through the presentation. But however, this is an understandable, valid question many of you could ask, especially we get this from our reviewer. She or he has the strong recommendation and would like to know how that come out. So what we're showing you here, the table on the top, is a recent publication. The author used SimSip version 15 and look at 16 compounds and to show how the prediction, in general, yes, if you look at the twofold AUC, it is quite reasonable. So compared to, but how that compared to the work from this working group, where we have a 26 set of the data, where if you look at the AUC prediction within twofold and 1.2-fold. So we did this comparison. And to be fair, and we also did additional work using the same 21 set of the clinical data, and we ran the simulation, either used the version 16 and also our built model, because at the time when we started building our model, we also used the SimSip version 16 as the base to build upon. So the graph basically is showing you this 21 set data of the comparison, where, as you said, as you can see, right, without a comparison, we will see, yes, in general, not too bad, but as what we, but we do see the trend of a tendency of over-prediction with the existing model and so on. But again, I just want to bring you back, as I earlier mentioned, the value of the work is we try to get a more and more mechanism understanding, which will boost our confidence to use the model, simulate different scenarios. So that's turn to the last slides of our summary. So again, when we start this work, the recommendation of itraconazole DDI, mainly based on the literature review, which already provided a very good basis. But as that paper, clinical pharm paper recommended, the simulation used the PBPK model would provide a better or more confidence on those recommendations. So the existing model, when we started, mostly based on using a top-down approach, mainly due to the challenges getting the reliable in vitro data. So our work definitely show you that we using those newly generated data, and through our effort, we'll be able to build a better the model with a better mechanism understanding, which give us the confidence to use that to do the simulation and make recommendations. And overall, the recommendation from this work, I hope provide a useful information to farm industry as well as regulatory agency. And we think largely it to confirm the study design that proposed by the CPLG. So with that, I think we finished the part of our presentation. I think Majid will take back to further questions. Thank you.
Mahalia, do you want me to read some of the questions?
Oh, go ahead, Heidi.
Oh no, I just was wondering if you wanted me to read some of those questions that have been coming in.
Let's do that.
Okay, so Tammy and Yen, there's some questions regarding is the bioanalytical assay the same for all the studies, and how does that affect the overall conclusion? I think this came in when you're talking about the clinical data for the itraconazole and used in the model.
So yeah, no, we didn't, for the clinical data, most of the companies already had that data generated prior to the initiation of the work, so we didn't use a common bioanalytical assay, but I assume everybody's assays were, up to the standards required to generate quality data.
Great. Okay, thank you. And another question is, what is your opinion on using other stronger 3A4 inhibitors such as ritonavir? Maybe why we chose to look at itraconazole, as opposed to ritonavir, maybe?
Tammy, you want to see something?
Oh, okay. Yeah, I think itraconazole was one of the recommended very potent inhibitors, similar to ketoconazole. Ritonavir, I believe, is also very potent, but I don't know if it's quite as extreme of a case as itraconazole in terms of completely inhibiting CYP3A4.
Mm-hmm. Yeah, right. We realize, there's a published work also conducted quite a good analysis and talking about the option use ritonavir. I see some valid point there, but in my opinion, or our opinion, I think especially if, let's see, even from the model perspective, ritonavir could, in addition to have inhibition, it has the time-dependent inhibition. There could be some induction. So how to sort that out is a challenging, especially consider we're talking the clinical DDI study design. It probably will be more complicated that when talk about the design, especially with a different company, they could have a different substrate. So how you be able to have the design of those to right hit on the inhibition that you can eliminate the other mechanism in the data interpretation could be more challenge. That's my opinion. Yeah.
Thank you, Yan. I have another question. Could you please explain the reason why you chose not to include PGP inhibition in the model?
Yeah, I don't, well, first of all, I don't know that we had a good way to do that mechanistically. And the main purpose was to be able to predict CYP3A4 interactions and inform a clinical study design. So we decided that wasn't necessary for our purpose.
Okay. Lots of questions here. Okay. What about impact of CYP3A5 genotype? Did we look into that?
No, we actually didn't look into that. And also, I don't think that we would have had the correct or necessarily all the information we needed on the CYP3A5 FMs for any of the victim drugs.
Yeah, for both, yeah, just quickly add, I think for both the PGP and this 3-5 genotype are really good questions. So, but to actually build a model with good confidence, we do need to have the good data, relevant data, appropriate data, which we don't have it now. So, however, we hope the model we built already provide a good base that have covered all existing data and knowledge, so that people can use that build up upon when we getting more data, such as the PGP kinetic data, right? If we want to include that, we do need this. And there's a different reported number and the translation of PGP from in vitro to in vivo, if I remember, at least like a 10x different. You need to bridge in the data in order to capture the in vivo. So as such, I think those science continue evolving. We would be happy to incorporate that when more proper data becomes available. But we hope the model we already provided to have the good base that people can build upon that. Thank you.
Thank you. Another question is, I was interested to know why a hepatic uptake scaler was used for this model instead of building in transporter Ki values from in vitro data. Maybe transporter.
Yeah, so that's another one that we wouldn't be able to translate directly from in vitro data. So building in transporter Ki's wouldn't.
Yeah.
Are not something that have good IVIVE that you could build up in a bottom-up manner. So then we employed the hepatic scaler to recover the observed data, and the data that we did have was in the same range as in terms of the amount around the amount of hepatic uptake we were seeing. So it did make sense physiologically.
Yeah, thanks, Tammy. Yeah, that's exactly what we did. So in this case, we're thinking itraconazole, the substrate of the hepatic uptake. So the such as Jmax, Vmax, Jmax, Km, we don't have such data. So we definitely would welcome to see more those information and data, so we can further expand to have that part also more mechanistic. And a current input just based on the, I would say more of qualitative, even there's data such as the KPUU tested in the animal and also from in vitro in the human indicate that. So we don't have a very accurate number that also we think will be easy to translate, but we wish we could. If anybody have those kinetic data would like to try, that will be also welcome. Thank you.
Okay, thank you. There's some more questions. Could you comment on the solution versus capsule? Solution fasted seems to give larger effects than capsule fed almadazolam. Could that be partially attributed to absorption enhancers in the ITZ solution, and could that affect other substrates as well? That is not due to increased ITRA exposure, but increased substrate exposure from absorption enhancer is not related to itraconazole, CYP inhibition? It's a good question.
Yeah. I mean, it probably depends on the substrate as well, I would think, but I think the oral solution also has a little bit higher exposure than the capsule and the capsule needs to be in the fed state to have the highest exposure. There could be some differences there.
In the paper, we provide both models for solution and capsule.
Yeah.
Okay, question. Do you think the M-atom model would provide an improvement in DDI prediction in comparison to the first order or atom model? Secondly, was sensitivity analysis used to test the parameters for any mechanistic or systematic errors?
Yeah, I mean, that's a good question. As I, when I was mentioning when we built the atom model, we did have to add that scalar to the effective permeability. I don't think I'm not sure if the M-atom model wasn't available at the time. It may have been, but yeah, we didn't explore that to see if it better recovered the observed data with the bottom-up approach.
Yeah, just to add on that, I think that could be twofold of a different impact. I guess that's what the attendee is asking. Yeah, I'm sure. I think at that time we developed the M-atom model wasn't available yet. But if we think about it, that could be two things, right? One is we know the current in vitro data not be able to just predict the absorption. Mainly, we see that the permeability data need to be scaled up a lot in order to capture that. So what is the mechanism? Maybe M-atom model would predict absorption better from PK perspective. I guess the second level question is on the DDI, right? Whether that will change the predicted DDI in the gut. That could be also tested. So we haven't, we cannot directly tell if that must be changing or impact, but it will be interesting to test that.
Thank you. So another question is, how are the 19 sets of data generated by the member companies compared to each other? How did the working group handle the differences?
Mm-hmm.
So, I mean, I can actually show those plots. Yeah, I mean, you can get a sense of how they compare to each other because they're just overlaid here. So I would say there is some variability, but there's also a lot of overlap. And, obviously we group together the similar study designs, so only have the same study designs overlaid here versus slightly different dose regimens. But yeah, I would say, like, in general, it reflects the variability you see with itraconazole. I didn't think there was any.
It's related to another question someone had. How do you fit the itraconazole concentration in the different trials that show different itraconazole PK at the same dose? So probably that's a variability in the studies. The different.
So that one I'm not sure. So what? Can you repeat the question, Heidi? I'm not sure what you're talking about.
How did you fit that? How did you fit the itraconazole concentration in different trials that showed different itraconazole PK at same doses?
So yeah, we didn't actually do anything like that. So we just used the data to compare with our model to assess whether our model could recover the observed data. The data used in model development were IV data in the literature, and then some single and multiple oral dose studies. And the only fitting that was done were the distribution parameters, some of the distribution parameters from the IV dose And then the things we just talked about, like the uptake scaler, which was for the clinical data for that was using clinical IV data, as well as the oral data for the ADAM model. So I don't think that really came into play with all the different studies from the working group. And the data in the literature is pretty consistent with one another.
One question is, timing of midazolam administration might be different. However, in case of a victim, which is a substrate for 3A and PGP or BCRP, timing of administration relative itraconazole could have an impact. Can the speakers comment about that?
Yes, I think theoretically it could. But our analysis, what we can tell is mainly for the enzyme part, CYP3A. So yeah, I think that's all I can tell. At least the itraconazole study, the purpose in the clinical now still try to identify or confirm your NME is the substrate of CYP3A. And so the study design would be useful for that purpose. If you, with that design to confirm the CYP3 contribution probably is the most appropriate. But if the transporter, if the compound is a substrate of transporter, and that's in my own opinion, you probably not be able to make a conclusion based on just based on itraconazole study. And of course, this design won't be the best or appropriate. You probably have to conduct other study with other transporter, more selective transporter inhibitor to confirm that part separately, then put data all together to sort this. So yeah, this work, the design of this won't tell you that. I agree. And in theory, there could be, in fact. But if that happened, I think other study need to be added to deconvolute the different effect.
Great. Thank you so much. Thank you to Yen and Tammy. We have run out of time. Should I, we will maybe take care of these questions offline? What do you think, Mahalia?
I think that's a great idea. Thanks, Heidi. We'll go ahead and submit the remaining questions to the speakers, and they can respond to them as a group. Once again, I'd like to thank everyone for joining us today. I think it was a great presentation. I hope that you all enjoyed it. As a reminder, the slides and the recording of the webinar will be shared for IQ members on IQ Connect, and we will also share a version of the recording and the slides on the IQ website. I'd like to once again thank the speakers and Heidi for moderating, and I hope that everyone has a great day. Thanks very much.